Cell adaptation, which refers to the reversible changes a cell can undergo in response to changes in its environment. Here is a breakdown of the points listed. Size. This refers to changes in the size of a cell. When a cell adapts to increased work demands or stress, it can grow larger, a process known as hypertrophy. Number. This involves changes in the number of cells in a tissue or organ. An increase in cell number is known as hyperplasia, and this can also be a form of adaptation to meet increased functional demands. Phenotype. The phenotype of a cell includes its specific characteristics and functions, which can change in response to the environment. Cell adaptation may result in a cell changing its phenotype to better suit its new conditions. Metabolic activity. Cells may also adapt by altering their metabolic activity. This could mean increasing or decreasing the rates of certain biochemical processes to cope with new demands or shortages of resources. These adaptive changes are typically reversible. If the stress or demand is removed, the cells can return to their original state. Hypertrophy. Definition: hypertrophy refers to an increase in the size of individual cells, leading to an enlargement of the organ or tissue, without an increase in the number of cells. Stimulus. The primary stimulus for hypertrophy is increased stress or workload on the cells. This can be due to physiological stimuli like exercise, in the case of muscles, or pathological conditions like high blood pressure, in the case of the heart. Mechanism. The mechanism underlying hypertrophy involves the increased synthesis of structural proteins within the cells. These proteins include components of the cytoskeleton, enzymes, and other cellular structures that allow the cell to function at a higher capacity due to the increased demand. Tissue response. Tissues that undergo hypertrophy are typically those that cannot divide. This means that the cells in these tissues have exited the cell cycle and do not go through mitosis. Examples include cardiac muscle cells and skeletal muscle cells. Understanding cardiac muscle hypertrophy. Mechanical stretch. When the heart works harder, such as when you have high blood pressure, the muscle fibers are stretched more than usual. This stretching is a direct result of the increased workload. Molecular signals. This increased stretch, along with other signals such as hormones, like angiotensin, and growth factors, like IGF-1, sends a message inside the cells that they need to grow. Activation of transcription factors. These signals lead to the activation of special proteins called transcription factors. In the heart, key factors include GATA4, NFAT, and MEF2. These factors work like switches that turn on certain genes inside the cell. Increased synthesis of contractile proteins. Once these genes are activated, the cell starts making more of the proteins that help muscles contract, like actin and myosin. Effective contraction. With more contractile proteins, each contraction of the heart muscle is stronger, helping to pump blood more effectively against the higher pressure. Induction of fetal gene expression. In addition to making more contractile proteins, The heart may also start using genes that were originally active when you were an embryo. For example, certain types of myosin, a motor protein involved in muscle contraction, that are used in fetal hearts might be produced again. This can be part of the heart's effort to work more efficiently under stress. For example, the atrial natriuretic factor, ANF, gene, which is active in the heart of an embryo but usually inactive after a person is born, becomes active again when the heart muscle enlarges. ANF is a hormone that makes the kidneys excrete more salt, which in turn lowers blood volume and pressure. Turning this gene back on helps to reduce the overall stress on the heart and improves its ability to pump blood. In hypertrophy, morphologically, the affected organ appears larger in size when observed with the naked eye. Under microscopic examination, there is an enlargement of both the cells and their nuclei. Hyperplasia. Definition: hyperplasia is an increase in the number of cells in an organ or tissue. These additional cells are a result of cell division, which leads to tissue growth. Stimulus. The stimulus for hyperplasia often involves increased workload or demand on the tissue. This could be due to a physiological need or a response to an injury. Mechanism. The key mechanism behind hyperplasia is cell division, specifically through the process of mitosis, where one cell divides to produce two new cells. Tissue capacity. Hyperplasia occurs in tissues where cells have the capacity to divide. This distinguishes it from hypertrophy, which involves an increase in cell size rather than number. For example, if a part of the body needs to handle more work than usual, like the liver after part of it has been removed, the remaining cells will start to divide more to make up for the missing tissue. This is the body's way of adapting to ensure that the organ can still do its job effectively. Endometrial hyperplasia. This condition involves the thickening of the endometrium, which is the lining of the uterus. It is often caused by an excess of estrogen without the balancing effect of progesterone. This imbalance can lead to a proliferation of endometrial cells, causing the lining to grow thicker than normal. If left unchecked, endometrial hyperplasia can increase the risk of developing endometrial cancer. Benign prostatic hyperplasia, BPH. BPH refers to the non-cancerous enlargement of the prostate gland. It is commonly associated with aging and influenced by androgens, male hormones, particularly dihydrotestosterone, DHT. This enlargement can compress the urethra, leading to urinary symptoms like difficulty starting urination, a weak stream, or frequent urination, especially at night. Some forms of hyperplasia have a heightened chance of developing into cancer. A endometrial hyperplasia can potentially evolve into endometrial adenocarcinoma, a type of uterine cancer. B. In the liver affected by cirrhosis, regenerative nodules can transform into hepatocellular carcinoma, a common type of liver cancer. Metaplasia explained. Basic definition. Metaplasia is when one type of mature cell changes to another type. This change is a response to stress and helps the tissue adapt to new, often harsh, conditions. How it happens. Stem cell reprogramming. In our body, stem cells can turn into different types of cells. In metaplasia, these stem cells are reprogrammed to replace the existing cells with a new type that can better handle the stress or irritation. Common causes. Irritants. Chronic exposure to irritants like gastric acid or tobacco smoke can trigger metaplasia. Barrett esophagus. Here, the cells lining the esophagus change due to constant acid exposure from the stomach. Respiratory tract. In smokers, the normal ciliate columnar epithelium of the airways changes to a tougher, stratified squamous epithelium. Potential risks. Progression to dysplasia and cancer. If the irritating condition continues, metaplasia can progress to dysplasia, a more disorganized and abnormal cell growth, which can further lead to cancer. Example. Barrett esophagus can lead to esophageal adenocarcinoma. If not treated or if acid exposure continues. Metaplasia in connective tissue. Myositis ossificans, dystrophic calcifications. This is a type of metaplasia where bone forms within muscle tissue, usually after an injury. Cellular adaptations, atrophy. Definition of atrophy. Atrophy is characterized by a reduction in cell size, cell number, and the amount of organelles within cells. It results in the shrinkage of the affected tissue or organ. Types of atrophy. 1. Physiological atrophy. This is a normal process where cells reduce in size or number as part of the body's natural growth or aging. Examples include the involution of the thymus after childhood and the notochord after embryonic development. 2. Pathological atrophy. Occurs as a result of adverse conditions and can lead to the diminished function of the affected tissue or organ. This can be caused by disuse, reduction in workload or activity, leading to muscle atrophy. Denervation, loss of nerve supply, often seen in paralyzed limbs. Diminished blood supply, ischemia can lead to tissue atrophy. Poor nutrition, inadequate nutrient supply can shrink tissues. Pressure, prolonged pressure can cause tissue atrophy. Mechanism of atrophy. Atrophy involves a decrease in protein synthesis along with an increase in protein degradation within cells. The ubiquitin proteasome pathway is often implicated in this process. In this pathway, proteins are tagged for degradation by a small protein called ubiquitin and subsequently broken down by proteasomes.